I'll take Q&A after each speaker, and at the end of the whole session, I'll have a collective sort of Q&A about sort of the bigger lessons learned. Our first two speakers are here with us in the room, and the second two speakers will be joining us virtually. The details of the bios, I think you know, are online and with the program. So I'll only give a sort of a summary introduction to colleagues. But the first speaker then is Professor Richard Lessels. And in this capacity today, he's based down here in UKZN. He's a outstanding academic, but he's talking on behalf of the South African National Advisory Group on Immunization. We call that NAGI. But for those of you familiar with WHO speak, that's the South African NITAG. I've worked with Richard on a number of things. He and I have worked together. It's been always a pleasure to work with him. Hugely insightful and to the point, and stops the rest of us having frills when we talk. So he's going to talk about South Africa's approaches to ensure access to vaccines during public health emergencies. But I'd very much encourage you then to think about what the questions are as he talks, because I think many of these topics that you're going to hear lend themselves to discussion. So as Richard talks, do think about that, and we'll have Q&A straight afterwards. So Richard, over to you. Thank you.
Thanks so much, Helen, for those kind words. I'm not sure how many of them are true, but they're very kind. So yeah, I'm just going to say a few words to kick off this session, and to try and bring some insight from the South African perspective, from a NITAG perspective, about thinking about vaccines during public health emergencies. But I hope that we'll spend the bulk of the time on this panel discussion. It's not progressing. Yeah. So just a reminder that we are all working towards this immunization agenda 2030, and most of our countries are signed up to this immunization agenda. And you'll be aware that one of the strategic priorities within that immunization agenda is outbreaks and emergencies. And just to go through the wording here, the goal is that immunization programs can anticipate, prepare for, detect, and rapidly respond to vaccine-preventable and emerging disease outbreaks. So really a lot about what Helen presented to us this morning in the plenary about the need for strong surveillance, the need for strong clinical diagnostic capacity, so that we can understand what's happening with emerging, re-emerging diseases, vaccine-preventable diseases. But the other thing to highlight here is this is, of course, also happening in the context of much broader changes to immunization programs and immunization strategies. And at the same time, as we're thinking about pandemic prevention, preparedness, we're also thinking about more routine vaccination across the life course, and thinking about introducing many more vaccines for adults, for pregnant women, for people living with HIV, for other key groups within the population. So that provides a challenge for us when we're thinking about the framework for prioritizing vaccines, for introducing new vaccines, and for preparing and preventing pandemics. We've talked about this this morning, and Loazi first mentioned and then Helen highlighted. We really can never forget this problem with the COVID pandemic and the inequity in access to vaccines. And whenever we look at slides, we show different slides that show us some of the numbers about this, but we always have to remember back to the human suffering and what we all experienced with colleagues, with family members, with friends dying due to delayed access to vaccines. And the key thing for me here has always been not the huge gap that you see at the end, but the gap that you see at the beginning in terms of the speed and the ability to rapidly get access to vaccines where the need was greatest. And that's something that we are still now, of course, seeing repeated. And so coming to prioritization, this is something that I find quite overwhelming as a clinician, as a clinical epidemiologist, and as someone who has very broad interests in lots of infectious diseases. I find it really challenging, and why I'm looking forward to the discussion today is to really drill down on how we think about prioritization and how we make decisions and make those decisions collectively. Helen talked, and I think one of the later speakers in this session will talk to the WHO list of priority pathogens, of families, of prototypes. This is another example on the right from the Africa CDC, where they also did a prioritization exercise to try and guide decision-making around priority pathogens of epidemic and pandemic potential. And so we see many of these exercises at lots of different levels in our global health infrastructure. And we have to think and discuss how we align that prioritization process from a national level to a regional level and a global level. And it can be overwhelming because, as Helen also highlighted earlier, we have this increasing potential for emerging and re-emerging diseases. And on the left is just an example map showing some of the main outbreaks and some of the main diseases and some of the kind of upsurges in vaccine preventable diseases across the continent. And here's the challenge, because again, we highlighted this morning the huge disparities in our understanding of some of these diseases, and particularly across large parts of Africa, our weaknesses in surveillance, our gaps in surveillance to really understand the epidemiology and the burden of disease associated with some of these diseases. And that, of course, is coming into the forefront now with Mpox, but it's the same with many of these other diseases. We have a very poor understanding of many of these pathogens, such as dengue, chikungunya, Zika virus, which are on all these priority lists. But our understanding of the current burden of disease across Africa is really quite limited. And so, again, feeding into decision-making around prioritization and also anticipation is really challenging when we don't have that core understanding. And so just to come to the current example, and really this was a test for South Africa to see what we'd learned from the COVID pandemic and whether we had the systems and structures in place to respond more rapidly when a new pathogen entered the country. And all of you will be aware that although this is somewhat indirectly related to what's happening in Central Africa, Mpox started an outbreak here in South Africa at the beginning of May. And here I'm just showing you the epidemic curve for the Mpox outbreak, and you can see a small number of cases. So still to this day, just 24 cases detected in South Africa, but amongst those 24 cases, 3 deaths. And you can see that the pattern of this outbreak followed very closely the global outbreak that began in 2022. And we have an understanding that this current outbreak is related to the clade 2B virus. So we saw that as a test case for how rapidly could we respond, given the uncertainty about how this outbreak might evolve within South Africa. And so we very rapidly formed structures within the NAGI, within the National Advisory Group on Immunization, a technical working group which Helen and I co-chair, and very rapidly produced some evidence-based recommendations to the Department of Health around deployment of vaccines to control this outbreak. And all the mechanisms and all the structures and the systems were in place to be able to get emergency approval for vaccines and to have a plan for deploying these vaccines to the people at highest risk in South Africa. But just last week, we passed 100 days from the first confirmed case in this current outbreak in South Africa, and we still have no vaccines in the country to administer to people at risk of infection and to people at risk of severe disease and death from Mpox. So this has still a learning curve in terms of the processes and the systems for rapidly obtaining vaccines and deploying for a public health emergency. And one of the things that I want to hopefully discuss more as we open this up is the risk that we also, the risk of complacency and the risk of deprioritization. And when we experience outbreaks, we sometimes still again get a false sense of security about what's happening. And so when we think about the South African Mpox outbreak, I show you here that this seems a relatively small outbreak. And yet, if you look at recent modeling, the modeling suggests that probably there have been somewhere between 300 and 600 infections over the first few weeks of this outbreak. So again, because of challenges with surveillance, with case detection, with awareness, we can have a misunderstanding about how epidemics are progressing and the potential for escalation of epidemics. And that risks then a complacency and a deprioritization if we have that weak understanding of surveillance. So I just wanted to finish there and say that we really have to come together and think about how we do this prioritization and how we align at different levels of the global health architecture, and how that feeds into decision making about vaccines at all these different levels. So I'll finish there and open up for any questions. Thank you.
Thank you. So are there questions there? This was very much looking at a country response. Are there questions from the floor on this? Not seeing any. Oh, there we are. Yes, please. Do we have a runner here or a microphone? Thanks, just over here.
Thanks. The name's David Jarvis from the Cedar Labs. Do we have any wastewater surveillance implementation in South Africa for an increasing number of pandemic threats like Mpox? I see that in the US they are implementing it. Our COVID-19 wastewater strategy seems like the only monitoring we're doing right now for COVID-19. it seems like the logical choice to go down that route.
Richard.
Yeah, a really good question. And there has been a lot of expansion of the wastewater surveillance and the genomic surveillance linked to that as a result of the capacity that was built up during COVID. And the NICD, the SAMRC are trying to really capitalize on what was learned during COVID to expand that to other pathogens. Of course, we have to, again, have a core understanding of how useful that can be, and it has to be able to inform decision-making again. I can see the use case for wastewater sequencing for some pathogens, but for others, that will be much less. So I think that certainly we have built the capacity and we need to use that. But a lot of the wastewater sequencing is still coming through research funding because of interest in this kind of emerging field rather than kind of core funding for ongoing public health surveillance. So we're going to have to present the evidence that this is something that can benefit and should have sustained funding and support in the long term.
Yeah, a very good question. And I'm sure people are aware that in Gaza, there were 4 cases detected environmental samples of vaccine-derived polio, followed then almost inevitably by a child with flaccid paralysis. So I think that this is, you also, as Richard said, you then have to know how to interpret it, but then how do you use it? And I think that that's going to be something, it's an emerging field, but it's a very important question. Other questions? Richard, could I ask you, I mean, what should be so I'm going to put my researcher hat on and working with NICD and working with vaccines, and we've immediately said, well, we need to do some vaccine research to look at the safety and effectiveness of the MVA vaccine, which is the one we would hope to get in here for people living with HIV. And then with NICD, we want to do sero surveys of risk populations. But it's not immediately obvious who's in charge. I mean, it seems like, people will put their hand up and say, I want to do this, but should there be better coordination? And the other thing is, where do we get the money from to do this?
Yeah, I mean, I'm sure that you and I agree that there should be better coordination. And again, I think that we've fallen back a little bit from what we learned of the real strength of South African science coming together collaboratively during a pandemic. And really that was because of lots of leaders like Helen in the country bringing together everyone and all the different research groups together with the public health bodies and all the other stakeholders to really drive the research forward collaboratively. And I think that we're not seeing that same collaboration and the same urgency with mpox. And you're quite right, there's clearly very limited funding in the global health arena for mpox, and most of that is now, of course, being directed to the areas with the highest, with the most urgent problems. So getting funding for that kind of research here in South Africa is clearly very challenging. But I haven't seen the same urgency with this outbreak as we saw, and so I think we're falling back into bad habits of working independently on the same problems, and again, not learning lessons.
Okay, so I'd like us to hang on to some of these thoughts because some of them are going to cross-cut across all of the speakers, and clearly one of them is thinking about how do we raise these funds quickly. and as a global and a regional collective around these matters. So thank you very much, Richard. We'll go on then to the second speaker, who's Mr. Ondari Mogeni. He's the Global Affairs Director at IVI, which is based in South Korea. It's the International Vaccine Institute. Many of you will be familiar with it. And he's going to talk about IVI's strategic goal to support pandemic preparedness. So over to Ondari. Thank you.
Thank you. And I'd like to extend my gratitude to Dr. Tommy for the invitation to be here today. And essentially, I'm going to speak to you about IVI and how we think we can contribute to pandemic preparedness as it's been said that the next pandemic will be around the corner sooner than we thought. And I thought the best way to go about it is to tell you who IVI is, because that background builds to what we are currently doing and the plans we have going forward. And the International Vaccine Institute essentially is an offshoot of the United Nations Development Program, founded in 1997. And at the time, the mission, which still remains the same, the mission was to discover, deliver, and develop vaccines to low and middle countries, largely and incentivize vaccines that big pharma might not be able to want to tackle. And I think when we were founded, Korea government elected to headquarter the institution among the 6 bidders. And we currently are headquartered in Seoul with a regional office in Europe, in Sweden, then also a regional office now on the continent in Rwanda. And essentially, we have about 43 member states, including WHO, who are state parties and signatories. And how we work is that we have the privilege of working through the entire value chain, from discovery all the way to delivery. And when you go through it, various components in each category. And why I put this up here is essentially, on one hand, we look like an international organization, which indeed we are, because we are treaty-based. And on the other hand, we do a lot of basic research at the lab level and preclinical work. And then also on the other hand, we work like a PDP, doing a lot of vaccine development, epidemiology and surveillance, and ultimately all the way to delivery. And that's how that builds up to be able towards our mission. And I wanted to bring here that I think he rightly pointed out some of the lessons we learned during COVID, then we sort of are forgetting about them. But I wanted to look at this slide that in the same way where we were about, Korea was about like, I think almost now 15 years ago, during the H1N1 pandemic, when they did receive vaccines from one of the governments, but the condition at the time was that should we want those vaccines, we are more than happy to come for them, what we really experienced again 4 years ago. And I think as a result of that, the current government purposed to then grow the vaccine ecosystem to not only be 80% vaccine self-sufficient, to be able to be the number 5 producer in the world. I think right now they're still on course to that goal. By 2030, they're about 50 to 60%. But why I'm showing this slide is that as IVI, we have been at the privilege of being able to work with them across all the stages, whether infrastructural development, the skill sets development, to currently one of the initiative K-Hub that I'll be able to talk about later. And you'll be able to see like Biologics that currently is contributing to 90% of the current global stockpile for OCV. At the time when we wanted to started working with them was a very small biotech, but I think now significantly contributing to public health. We do this work with a lot of people, so I'll be able to skip that. But then he showed earlier also a slide on COVID, but I wanted to show this one to indicate that I think when you look at those metrics, the Africa versus the world, you realize that whether the people who had complete vaccination, the people who had at least 1 dose, or even currently those who've been able to get a booster dose, there's a huge discrepancy when you look at the entire globe, but also when you look at Africa, and in some cases even less than 1%. Of course, then this leads to some of the thinking post-COVID, and that is how, for instance, PAVM, which is now FARM, came about with the ambition that we should move from the less than 1% that we do have, hopefully by 2040, to be able to produce up to about 40 to 60% of our needs. And in that thinking, we felt then how can then we be able to contribute given the amount of work we do, because across all the more than 50 countries across the globe. And we came up with this idea of advancing vaccine end-to-end capabilities in Africa. This initiative coming from the background that I think we have the unique opportunity of being able to work across the value chain. And as Abebe earlier mentioned, I think you do have almost more than 20 governments that did announce that they want to do vaccine manufacturing, but it's one thing to have the plant or the infrastructure in place, but there are many things that feed into it. You need the drug substance, you need the regulation, you need the R&D, you need the market demand. So, and all those have to work in tandem if it has to be sustainable at the very end. And this came out of the leadership that they felt that there's need, because we have largely worked out of Korea, but they felt there's need now to have that physical presence. And of course, that led to the regional office being opened up in Rwanda that largely will lead our diplomatic efforts all over the continent, and more specifically this AVEC office that will be a project office to oversee our vaccine continental efforts that will be based in Nairobi. And I think we believe by so doing, as I mentioned earlier, by contributing to the farm objective, we can strengthen pandemic preparedness and ensure that I think we can be able to control and prevent endemic diseases and improve continental security. And by virtue of the uniqueness that we can be able to work across the value chain, then we can plug into the and leverage on the already existing efforts on the continent, be it in epidemiology, be it in tech transfer. And I'll briefly talk about later some of the tech transfers numbering up to 13 that we've been able to do, and ultimately ensuring whatever vaccines that are produced are delivered and accessed through generation demand. FARM did come up with a framework of action that has almost 8 strategic pillars, and we do believe that we can then be able to contribute to 4 of those, because I think Africa CDC through FARM, which has that member state mandate to be able to oversee vaccine biomanufacturing on the continent, but we see that framework from the top-down approach, and I think we see the partners. Then there's an opportunity to contribute to its implementation through projects and across the value chain from end to end to be able to realize it. And in that way, we see that roadmap that I think for it to be successful, then R&D is important. You need to have technology transfer, whether of priority pathogens, and holistic technology transfer that works with those respective recipients to ensure that I think whatever is transferred actually is produced. Then, of course, regulatory authorities have to be enhanced. It was earlier mentioned we now have at least maturity level 3 authorities on the continent, because then they have to work in tandem. And the reason I'm saying like this, we did a technology transfer, for instance, to Incepta in Bangladesh. And of course, they've successfully went through, produced it, and went through the national authorization. But because of lack of their regulatory authority being maturity level 3, I mean, it couldn't get WHO PQ. So they do have a product that can be able to be utilized, but that can only happen nationally or through bilateral engagements. But at least the way Biologics is contributing to global stockpile of what is already a limited vaccine, that cannot be able to happen. Then, of course, there's the actual manufacturing. But as my director, Dr. Jerome, usually mentions, it's one thing to have the access to the vaccine, but if it's actually not delivered and utilized, then it's less meaningful. And in this case, I want to give an example of some of the things we do, that we have the ability to do a lot of preclinical work, and that's how we supported most of the partners during COVID to ensure that at least a lot of preclinical work was done during the COVID period. We also were able to develop a neutralizing assay. So we have a robust team that can be able to do a lot of preclinical work. And the other one is training. And here I highlight some of the trainings that we do have. Through WHO and Korea Ministry of Health for the last 3 years, we've been running a training on biomanufacturing. We call the Global Training Hub or Biomanufacturing, that we do hope to scale it up, while you see in the later slides, up to 2026, 27 to be able to have actual wet practice. And the other one we've been running is the vaccinology course, which might be of interest to some of us, as you'll see in the I'll go forward in the next slide, then I'll come back, that we are running this year. And the uniqueness of it is that while it has always been run out of South Korea, the headquarters, we tried last year simultaneously in Sweden and Korea. And then this year, we also wanted it to run it simultaneously in Korea, Kigali, and Rwanda, because we believe there's need for capacity building on the continent to be able to provide the much needed skill sets. So I wanted to put this slide that fills into the other, in the sense that both the OCV that currently is being utilized, as well as was developed in-house with technology transfer. The one thing that we do not have is the mandate to manufacture. So we basically did the technology transfer to industry, of course, with the commitment that the production should be at cost to be available for public health. And then number two was typhoid conjugate vaccine. And the uniqueness about this is that they're all been able to go through different processes that contribute to clinical development, pre-qualification. So there's a lot of capacity building that goes through the value chain. And that vaccine number 3 now, it's INTS and Shigella. But I want to give an example of the cholera vaccine journey, which has been a 20 years journey that we have had as IVI. And what I will want us to focus on is the top, where it indicates across the entire value chain from epidemiology to access, but also at the very bottom. And you realize that across the value chain, we've been working with different partners. And then I'll go back to, for instance, Biologics, as I mentioned earlier, who, when we started working with them, they were basically a small biotech that was starting to build to bad. But right now, they are the only ones currently contributing to the global stockpile of the much needed vaccine that is in limited supply. And of course, we have also done others to India, but also I'll mention in the next slide what we also are working on the continent. And I'm showing this looking at Dr. Morena, because he's the one who can speak more better to this. But this has been a privilege also to contribute to vaccine availability on the continent. And these are a technology transfer that began almost 2 years ago. But one of the beauty of it is that since the technology transfer began, working together with the team at BioVac and also at IVI, and right now with the scale up being done locally, and I stand to be corrected, but I think moving now into clinical development. And it's a journey we do hope we can be able to walk through with the relevant partners, primarily led by the South African colleagues, be it from BioVac, be it from SAPRA, to ensure that it can be able to achieve WHO PQ, and at least we have products that can be procured by UNICEF and Gavi, such that as a member state or African governments they can say, we want to procure this product that we do know has been produced on the continent. This also gives an example of other projects that we think can be able to then be led out of the AVEC Africa office. And the uniqueness of it is that some of them, like for instance, where there's a Starix, is a situation whereby we are part of the process, and probably later during the discussion, Petro can also comment about the mRNA, but whereby we're working with the partners, and we believe that's one way of growing the continental capacity. If ultimately, not only we should be able to be sustainable, but also we are able to deliver the mandate that there is of vaccine self-sufficiency. So I'll go back to COVID and basically say, and he rightly mentioned about leadership, that there are quite a number of things that we looked at COVID, but I think from where we look at a couple of inequities, be it lack of diagnostics, insufficient R&D, all the way to vaccination. And we believe if they need to be tackled before, if we can be able to be ready for the next pandemic. And underpinning all these, we strongly believe they need to be a coordinated and effective leadership. Because if we are able to do that, then it goes back to what IVI we believe on, relying on the existing burden and then being able to innovate and then ensuring that whatever has been innovated goes to commercialization, ultimately will be able to lead to the much desired impact for a healthier nation. And in that way, we can be able to be ready for the next pandemic. Thank you.
Thank you very much. So I think that's very interesting, because I knew that there was a plan afoot for the African region, but I hadn't appreciated that it had developed quite as far. So are there questions? I know we have colleagues here from CSIR and so on who might want to putting you on the spot. Are there questions that people would like to raise with respect to what we've heard, including this African initiative? Yes. Thank you. We'll get you a microphone.
I wasn't initially going to ask any question, but certainly I think there is a very huge opportunity with what has been presented here to localize technologies and avail them with the intent of the VIM strategy that we heard of yesterday with respect to looking at technology localization. And it's a conversation that we would like to have further in the public interest, obviously, to provide that platform that avails the pull-through of concepts, lend the technology and let the commercial companies pick it up, pick whatever is successful at early stage clinical or maybe medium stage clinical development and then produce it further. So very excited about the possibilities with IVR.
Would you like to say a little bit more about what you're doing or planning to do?
All right. So it is part of what we presented yesterday at the plenary session where we did the launch of VIMS. But effectively what it is building on existing capability that has been established by the National Treasury and the Department of Science and Innovation, which is to have small-scale capabilities and clean rooms to allow the system in South Africa to pull through products that come out of research and development, make clinical grade batches on multiple platforms, and then clinical grade batches allow for a scale that lets you do phase 1 clinical trials, clinical grade materials. So that's what we want to be able to do, to just be that bridge between the excellent research and development that we have and clinical development and manufacturing of product. And ours would be a dedicated capacity when you would find that commercial companies focus on particular products and see them through. Ours is to focus on facilitating the flow through the system from research, concepts and prototypes to clinical grade batches that then the innovators and entrepreneurs and commercial companies can decide if the target is worthy of further development. So we have that physical facility coming to completion this September, and then obviously we'll go on the long, hopefully not such a long road with Tumi to get it accredited, and then really find a way of building on that to develop potentially a contract development and manufacturing infrastructure for the system. So that's what that initiative is about. We hope it can be seen through, but certainly there's been a lot of investment already in the infrastructure. Hopefully we get more investment to expand the technology platforms that we can put on that facility. So thanks for the opportunity, Helen.
Thank you. Thank you very much. Other questions to IVI? We've just heard this exciting initiative for the African region. Other questions that... So I guess one question to you, you mentioned with the Africa's CDC, but one question would be, how are you going to work out your priorities? Because we're not short of choice for end-to-end development, but how are you going to work it out, given that there are multiple small initiatives, there are different manufacturers who are looking at early development of vaccines? How are you going to both fit into that, but also how would you work out your prioritization?
So I think one, in terms of the prioritization, I think that is largely coming, one, from the member states. And then secondly, I believe might have mentioned earlier, but the other exercise they've been able to do, they've been able to do an exercise that has prioritized about 8 antigens. And of course, the hope is that this then aligns to member states' priorities as well. I think that's one issue. But also secondly, is the realization that you might have some countries that might be able to manufacture, but then the others that can be able to fit into it, be it from capacity building, from some of the inputs. Because the reality is when you go through different countries, even you find there's one that's saying we want to manufacture as soon as tomorrow, but you find they only have a greenfield. Then when you talk to them, then the reality becomes alive that it's a long process. So again, naturally, and referencing what Abebe said, there are those who might be able to fall by the wayside in that actual process, but then together they can be able to work together to support each other. Because let's face it, on the continent, there are almost maybe 4 or 5 countries with a sufficient birth cohort that can sustain their own vaccination. And for any of the effort to be sustainable, then it has to be a pooled effort. But it has to be a continuous conversation, because then you can't dictate what the priorities need to be to the respective countries.
Thank you. I just want to ask another question, and actually we've got a number of manufacturers also in the room. You know, one of the things, I don't know if anyone else has ever gone on these courses where you do futuristic thinking. And the one thing that really occurred to me is that in 10 years' time, people are going to turn around and say, well, we can't believe that you had multi-dose vials and needles and syringes and cold chains and you had to mix things. We can't believe that you thought that was okay. So one of the questions is, especially given that, you know, in many contexts in our region, particularly if you're looking at outbreaks, you're going into very difficult, hard to reach zones. How much focus is going on from IVI on new delivery platforms, whether it's microarray patches, whether it's more stable products? Because we haven't really spoken as much about that as the idea that we need vaccines for outbreaks and emergencies.
And you're right, rightly so, delivery. So when I mentioned earlier the entire value chain, it has to start from discovery all the way to actual delivery. And in so doing, then you have to speak to those other aspects, like what you require. I may not be the right person to completely speak about it, but I do know my colleagues are working like the patch mode of delivery and other partners. So as time goes by, there's need to look at what are the efficient and effective technologies that we can be able to leverage on beyond what has traditionally been known. But I think still we can work with what is already there as we pivot into that, because at the end of the day, even as you want to make manufacturing a reality, given how capital intensive it is, how do you then initially, for instance, like whether even if it's monoclonal antibodies, whether it's the syringes that you've mentioned about, we still utilize tons of them to utilize for vaccine delivery. And I do know, for instance, now there's the company Revital in Kenya, in Mombasa, that has been supported. They're able now to meet the 50% of the needs of the continent. So I look at that twofold in terms of like thinking through new technologies that are going through the different phases of discovery or development, but also leveraging already on the existing technologies, because ultimately you want to bring the cost down if you want to make it sustainable.
Thank you. I mean, I think that this is just a whole area that we probably need to pay more attention to in the future, definitely.
Helen, Pietria, can I make a quick contribution?
Yes, please come
Thank
through.
you.
Yes.
Pietria de Blanche from Afrigen. Great that you introduce the concept of innovation in the discussions, and in the next session when we talk about manufacturing, Afrigen particularly will focus on innovation. Maybe just to share, we are working with IVI and Quad Medicine in Korea to actually demonstrate that mRNA can effectively be put on a microarray patch. So that innovation happening. And then this morning there was a CEPI announcement where we actually innovate now and looking at synthetic DNA so that we go cells-free for mRNA to drive the cost down and to support pandemic preparedness and speed of manufacturing. So in the next session, we will have some discussions on innovation in the manufacturing sector. Comment about IVI, great collaboration where if I may share, in 2022, in December 2022, when Afrigen was scale up and we had to take 22 people to 147 and train completely horizontally and vertically integrated the skills required for end-to-end research, development, and manufacturing. IVI sent teams out to Cape Town in our facility to train our people in our own facilities because we couldn't send them out of the country because of the pressure of work. The collaboration with IVI on the Rift Valley fever, I hope that there will be soon announcements around that, following a one health approach using mRNA technology, self-amplifying and modified to eradicate Rift Valley fever. So a great partnership that IVI is concluding on the continent with many, many of our partners. Thank you for that.
No,
Thanks,
that's
Professor
very
Petro.
helpful. Thank you. Did you want to?
And I was saying thanks, Professor Petro, for that contribution.
Yes,
Not much
yes.
addition.
But it's very good to actually hear that it's working, because it's one thing when you see the slides of what we want to do, but actually, and it works. I think that's very helpful. So I don't see any other hands. I'm going to then then move on. Nikesan Dembi from Africa CDC is tied up at the moment. I think poor old Africa CDC is really under pressure given the amount of media attention that's going to Mpox at the moment. So I'm not sure if he's going to try and rejoin, but I think it's unlikely. Anna Maria Restrepo is on the line from WHO HQ, and she's somebody who I've had the pleasure of working with for many years. And for those, many of you might know her, but has played an absolutely outstanding role in the development of the R&D side of vaccines. And in particular, with the Ebola outbreak, I remember going to a big international meeting at WHO where quite a number of role players were proposing what they were going to do to actually develop an Ebola vaccine. Some of it was so ambitious it would have taken 100,000 people to be enrolled. That's how ambitious it was. And Anna Maria and her colleagues, very much Anna Maria, one of the leading figures in this, came up with this very simple idea of ring vaccination based on smallpox. And that's this is the RSV, the RVSV vaccine that we now have. So I'm delighted that Anna Maria is here. I mentioned the pandemic, the pathogen prioritization, and she's going to take us through that in more detail. Anna Maria, over to you. Thank you.
Thank you, Helen. Just before I start, I want to say all that was a team effort, and you were part of it. So thank you for your continued collaboration. So I will try to share my slides and hope it works. So can you see my slides now?
Yes.
Yes. And now presentation mode. Does it work?
We're not in presentation mode yet.
And now
no, that's now you got the speaker mode.
Okay.
There we are.
Perfect.
Yes, you're perfect. This is good. Thanks.
Super. Thank you very much, and thank you to the organizers. And I'm so sorry I could not go, but we are super busy with the Mpox, and so I was unable to travel. But next time I will go for a bit of South African sun. So today I want to talk to you about the topic that, as the previous speakers have mentioned, is of the utmost importance to all of us. How can research contribute to the preparations for the next pandemic? So I will start with a bit of history. On the 7th of April 1948, the constitution of WHO was put into force, marking the official establishment of WHO. And one of the earliest research-related activities that WHO did after it was formed was in 1947. It was a service for collection and dissemination of data on disease outbreaks and health trends globally, providing the foundation to the informed public health decision-making processes that we enjoy today. In addition to that, the development of a new tuberculosis drugs in the 1940s and early 50s revolutionized the treatment approaches. Therefore, in 1950, WHO launched a mass tuberculosis inoculation campaign using the BCG vaccine. This was perhaps one of the first large-scale vaccination drives aimed at controlling a major infectious disease. In 1959, WHO established the TB Chemotherapy Center, later renamed as the TB Research Center in Chennai, India, to conduct research in tuberculosis treatment. I am saying this because we all have to remember that we come from a long history of working together to control diseases. So the second point is that together we have made substantial progress. Since the R&D blueprint was initiated in 2015, after the resolution signed by our 194 member states, including all of those from Africa, thousands of scientists, developers, regulators, experts, funders have worked and contributed to the development of tools for specific pathogens, roadmaps, target drug profiles, trial designs, regulatory pathways, and so forth. That is not the end of the story, but we have a lot to build on, and thanks to everyone, perhaps many of you in the room have participated and contributed to this. The next point is to talk about which pathogen will cause the next pandemic. that is the conversation today. So we are using a metaphor to discuss this, and we think the metaphor of the drunken man who lost the keys is very important. The first thing they will do is to look under the street lamp, because this is where they know they see the light. This represents for us the priority pathogens. Who are the recognized pathogens that some of you have talked in your presentation, because they are well documented, because we have significant efforts ongoing to control them. But knowing that there are about 1,700 pathogens that can infect humans, we could expand the light and prepare. And this is where the concept of the prototype pathogens come. Because if we work also on representative pathogens selected to serve as a model for basic and translational research to develop medical countermeasures that can be applied to any pathogen in the family, that will be very important. And all of you who knows the details of how the messenger RNA vaccines came to be so quickly understand that it was through this approach of using prototype concepts that we have the vaccine so fast. For example, understanding how the virus enters the cells, etc. Then there are the dark areas around where these thousands of pathogens are. Neglecting those areas of the pathogens that we don't have in any of our lists is unwise, because the next pathogen, the next pandemic pathogen could emerge from that. So therefore, we need to ensure that we understand what are these pathogens that we do not consider today a fake threat, a public health emergency of international concern, but that given changes in the biology, the environment, etc., could become one of them. This is why WHO is now proposing that in addition to the efforts that people have on priority pathogens, we should work on research across all families, regardless of the perceived public health emergency concerns that we have today, because our concerns are based on the data available today, and we cannot underestimate what will happen. Then we think that working on development of medical countermeasures for priority pathogens could contribute to sustainability of R&D in all parts of the world, particularly in areas affected by these pathogens. But we also think that working on the prototype pathogens, as I say, selected primarily to serve as representatives of the viral family or across functional groups, could help us very much to really be prepared for the next pathogen, because we know that uncertainty is the most important thing. The pathogens will not read our memos and our lists, and so we don't know which one will it be. So all this together is what will help us to prepare for the inevitable, that is the pathogen X. So moving forward, the priority families was evaluated with consideration given to all the pathogens that they encompass. This figure depicts the 29 families of viruses and 1 of 5 bacteria that were evaluated using the most recent ICB nomenclature. As you read, 1,700 pathogens were considered. The families were classified based on their capacity to harbor pathogens that we know now, at least one pathogen that can harbor a priority pathogen capable of causing a pandemic on the basis of our knowledge today. Of significance, 4 families were categorized of low risk by their own a small group of experts, and when we brought all the experts together, that was confirmed. These are the Annelloviridae, the Papillomaviridae, the Poliomaviridae, and the Herpesviridae. They were reaffirmed as low risk during the deliberations, but it doesn't mean that we should forget them. Therefore, we'll continue to monitor that. Of the families considered, the ones without endemic virus, the only ones without endemic virus are the Arena, the Borna, the Bunia, the Philo, the Rhabdo, and the Toga. So most of these viruses that we are talking about compressed in these families are endemic in one or more parts of the world. So if we only focus on the virus, we also, for each of the viral families that were priority, the experts identify the priority pathogens that should be the focus of our work. And here they are. And they are written with the standard current ICTV classification. If a family was considered to contain pathogens with attributes that suggest high risk, even remote, a prototype pathogen was also identified, and they are noted here. Selecting prototype pathogens is challenging due to the breadth of diversity between some of the viral families. Considerations of prototype pathogen selection varied across these viral families and included their importance as human pathogens, the current knowledge of replication and pathogenesis, the existence of animal reservoirs causing cross-species infection, and also the shared structural and functional properties of them. Overall, what underlines in everything is our current knowledge. So my thing is we should be humble and not arrogant that our current knowledge is still very limited. The good thing also to note is that the majority of the priority pathogens here highlighted in blue correspond to priority pathogens that we have highlighted in our previous reports. Okay, so now move. The priorities would differ only if regional perspectives are adopted. So if you want to go for only a piece of the planet, then you may end up with some differences. But as you can see in these two graphs depicting the priority pathogens and the prototype pathogens, many of these pathogens are present in all the 6 regions of WHO. Many of them are present in more than one region. So to stimulate research, it is important to have a locally relevant pathogen, but a narrow set of local priorities may leave us unprepared. For example, the arenavirus, Lassa virus, it circulates in Africa mostly, but there are the New World arenaviruses like Hunin. So there is a lot of merit in collaboration. Pathogens with worldwide distribution encompasses, for example, the subgenus Sarbecovirus or the influenza A virus, also some of the flaviviruses. And there are some prototype pathogens that are currently included in low-risk viral families, but they are present in all the 6 regions. For example, the human metapneumovirus and the rotavirus that we should keep an eye on. So here is a map to underline the importance of global collaboration in terms of research, the changes in science that should occur. As you see, here we show the mama arenavirus last science, and as I mentioned, this is mostly in Africa, but there are Hunin viruses and other viruses in the New World in South America, and same for the others. So we think that these distributions are often associated with the presence of animal reservoirs with the transmitting vector or with substandard living conditions. However, vector distributions can expand, pathogens can evolve, etc. This is why we need to keep a wider view and not just a local view. So moving on, there is a pipeline of candidate therapeutics and pathogens for the candidate therapeutics and candidate vaccines for those pathogens that were listed. I'm not going to go into the details, but just to show you the pipeline is not totally empty, but it could be additional candidates could be added. But the most important thing that we are saying is we are not promoting that we get a vaccine for each of the viruses that we listed and then put it into vials, but that we collaborate to have the knowledge across the family, to have able to generalize that knowledge and to apply it and be prepared for whoever the pathogen next is. We don't start from nothing, and the research and the R&D knowledge and the existing collaborations, many mentioned by the previous speakers, are invaluable. It's impossible to acknowledge all this invaluable progress, but we need to do more and we need to do better with a wider vision. So how do we move forward with this information? It's just this a report from WHO, and will it join the other reports that we have issued? So in terms of collaborative initiatives to ensure access to medical countermeasures, we have, some have underscored the importance of speed. But we in WHO believe that it's equally important to underscore the significance of access, equitable access, trust from the communities, and also safety. We also believe that it's crucially important to adopt a comprehensive perspective that acknowledges the paramount primary importance of all these values. And we think that it's critical that when we work on we focus on the science, because if we focus on the science and the technology, we can help resolve many of the problems. We have to be aware of the politics, but we should stick to the science. So before I talk to you about this collaborative open research consortium, that will be my final point today, I just want to tell you, I am from Colombia, but I became Swiss about 5 years ago. So I learned that unlike in other countries, in Switzerland, no one person is ever the head of a state. The president of the confederation is primus inter pares, first among equals. But with the office, there still comes a whole series of traditional duties and tasks. So with inspiration on that, in the proposed approach on how to implement the viral families, we are creating WHO collaborative centers that will act as the CORCs, the Collaborative Open Research Consortium. And this consortium will serve as the hub for the scientific R&D deliberations for that family. So they will not control it, they will not be the only ones who do it, but they will help us to move forward. This collaborative research worldwide coordination is imperative to bolster preparedness for potential future pandemics. Not politically correct, no opportunistic, but based on the scientific evidence we have of how this virus transmits, how they expand, and how we can learn more about how to protect humankind against them. So we think that employing a multifaceted research strategy is important, that engaging all researchers in affected communities through these collaborative open research consortiums for each family is also a way to ensure equitable global access and sustain adequate research capacity. So when we move forward in what these families, these courts will implement, the idea is that there is research in all the families with the smart surveillance, including, for example, wastewater surveillance that was mentioned, with research collaborative to establish, expand the knowledge about the viral structure, the infection mechanisms, the immune responses, and the host interactions. We also want to ensure that these courts help to work not only on priority pathogens, but also on the prototype pathogens, so that translational research is promptly used, that knowledge is promptly used to develop candidate products and vaccines that can be tested, that we develop common target product profiles and share mechanisms for development, that we have open discussions on choices of antigens and platforms, etc. Together, we can also work on help expand the research infrastructure, not only for clinical trials, but in general. And so that way, we can be prepared for the next pathogen. If we go in small networks and avoid the global networks of networks, we will achieve so much. If we work together, I think we will be better prepared. So in addition to the preparedness, like is happening now with monkeypox, we think it's important that the member states receive independent advice on which candidate vaccines or candidate therapeutics or diagnostics or other countermeasures should be considered for introduction and for evaluation in the context of outbreaks, that we work together to ensure that there are sufficient doses of these medical countermeasures available to be tested, like happens with Ebola, like we work courageously with 3 developers for Sudan Ebola virus in Uganda, etc., etc. There is a lot of discussion about clinical trials, core protocols are important, having agreements and funding, and a collaborative research at the base of that, in an open collaborative research, no closed clubs, no closed networks, but open to all the researchers who can contribute is what is required. So then finally, I just want to say this is not impossible. It's very possible, and I didn't have any alcoholic drink this morning before I talked to you. So why I'm saying this, we started with the Marburg consortium, that was a consortium for Marburg. All the developers researchers, etc., published a resolution to work together to advance the evaluation and introduction of Marburg vaccines. This was extended to all the filoviruses. And today, as we stand here, we have a research R&D roadmap, we have research agendas, we have landscapes, we have protocols, core protocols for vaccines and for therapeutics. And more importantly, we are engaged with 17 countries in the African region who are at risk of filovirus outbreaks, and we are towards getting the pre-approval of these protocols via the ABAREF network, so that the next outbreak, we are all ready, the researchers, the developers, the countries, etc. So our experience with and advice of the many leading researchers and institutions has reinforced the idea that this is possible, and we should work with all of you to make it happen. This idea reinforces that working on entire classes of pathogens rather than individual ones alone, that this approach can enhance our capacity to respond to unforeseen strains, emerging pathogens, zoonotic spillovers, transmission of animal pathogens to humans, and the hypothetical threat of pathogen X. This approach also aims to foster open international collaborations by establishing decentralized common reference points for researchers, developers, national authorities, and policymakers and institutions worldwide. I just hope, and we hope, that many of you will join the consortiums, and some of you will help us fill in the grid, so that we truly promote together, research in all families and R&D in the priority pathogens. In summary, we think that using this family approach and the proposed network of CORCs underscores the importance of identifying priority threats, but strategically working together to support the research across the families. We acknowledge that there are uncertainties about what pathogen will cause the next pandemic. This is why we are not betting on a short list of pathogens. We are working on to expedite global R&D efforts, and this is what all our member states have debated when we have the INB deliberations on the pandemic treaty in the Article 9. So this is really in response to what our member states think should happen, that we should have clear roadmaps, that we should have open collaborations, that we should work towards collaborative efforts, open and globally, so the equity in access to knowledge and innovations will replace the demands for equity in access of products in buyers and asking for percentage of those products. So overall, we think this collaborative and decentralized strategy seeks to support resource allocation to areas of research crucial for pandemic preparedness, ensuring the focus on these viral families and pathogens with the highest potential will help to maintain the research alive and funded everywhere, but we have to keep an eye on generalizability. And just to end, Helen, I just want to say that together is not an empty word. We want to thank the thousands of scientists worldwide who help us outline and develop this strategy. And I just want to say that we are convinced, and the scientists are also convinced, that if we work together, rather than in silos, we can solve this problem. Thank you.
Thank you. That was incredibly helpful, and I'm sure has given a lot of people in the room new information on how this is being framed. And I must say, when you were talking about collaboration, my mind went back. There's an excellent book, if you can ever get it online, on polio and the history of polio and the fight that went on in the development of polio vaccines. So a really good read, but the opposite to what is being proposed here. That was pure competition. So are there questions for Anna Maria? Anna Maria, can you stay online for a bit? Is Anna Maria with us? Yes, there we are. To me. Anna Maria, people will introduce themselves from the floor so that you know who you're talking to and what their portfolio is. Please go ahead.
Thank you, Prof. Rees, and thank you, Anna Maria, for an excellent presentation, and we appreciate you joining us albeit virtually, but really great insights. Two questions.
Just introduce yourself.
Oh,
Sorry.
apologies. Tumi Semete, I'm the CEO of SAPRA. So two questions to Anna Maria, and I've got one linked, but to Richard Lessels. I mean, this is a great, I think, strategic approach in terms of the focus from an R&D perspective, and understanding that a lot of the work in this area will be funded through grant funding. I wonder whether there's engagement with those that would be supporting R&D in this area so that there's a coordinated effort, again, from a funding perspective to align to this strategic thinking of priority families. And then the second question I have is, you know, we're in an era of data and know how powerful data is. Through these platforms, will data be easily accessible? I understand even under the pandemic accord, this is one of the sticky points around data sharing and access to data. And then a question to Richard is, with this framework that's being shared, which is really excellent, are African scientists aligned to this thinking process? Thank you.
Thank you. So let's take one set of questions at a time. We'll come back. We'll take those questions first, because I'll go to Richard as well. Anna Maria first on the funding side and coordination.
Yeah, I forgot to show my stellar slide at the very end. This strategy was launched on the Global Pandemic Summit in Brazil and has the full endorsement and support of CEPI. We also have approached other bigger players in the R&D arena, and we are getting a lot of positive feedback. So our aim is exactly as you say, to also get the possibility that when the courts identify the R&D roadmaps for a given family, that there is the financial support to implement the critical research that is identified, because that is absolutely necessary. But as I said, we start with a good point, and I want to thank here publicly Richard Hatchett and his team for all the support they have given us. Now, regarding, well, I forgot the second question, Helen. Can you remind me?
Timmy?
It's around data sharing.
Yes. So yes, I have been in the pandemic treaty negotiations, deliberations, and I know how that is an important issue for many of our member states. So what our experience is, not a political experience, but a research experience. We have now a global network for animal models and a global network for assays. And our experience is that when the discussion is about the science and how to get the science advanced, scientists all around the world are very keen to share even pre-publication data, information, etc. So we hope that this work that we do in advancing the scientific discussion will help encourage the parallel political discussions on data sharing and access. And then finally, I hope Richard says that the African researchers, well, because there are quite a number of African researchers already on board, that he will join us because we count on all your support.
Richard.
Yeah, well, I was going to say that. I mean, I think that is true. I mean, I think there's lots of positive developments there. That there's African leadership of these initiatives. We see it with MPOX, with the African MPOX Research Collaborative Network. So we see lots of development of leadership from the African continent, and I think that clearly we can always do more from that perspective. But on this, we're not being left behind, and we shouldn't be.
Thank you. Yes, go ahead.
Richard, yes, thank you. You know, I am a technical person and not the diplomat of WHO. So, and Helen knows it. I think it's very good that in all parts of the world, in Latin America, in Asia, in Africa, there are leadership initiatives. But my main concern, and our main concern, is that this turns into regionalistic approaches that then cut the possibility of global cooperation with researchers on the other side of the sea or up to the north or further to the south, because the focus is the nationalistic approach. So I hope we don't get there, and I hope the main focus is to ensure that the science and the equity in access to knowledge is what drives every discussion.
No, I think we would all agree with you. I think the other thing that we should look out for, though, is where academics are involved and pharma, is that almost by definition there is competition. So I think we're trying to change the sort of framework of thinking. So we're going to have to watch that space and really encourage that we have alignment rather than competition, which is destructive. We'll go to another question now. Thank you.
My name is Rachel Chikwamba from the CSIR here in South Africa. And my question is, I guess, to all panelists on prioritization of where developing countries, LMICs maybe, should place their efforts. The one thing that we want to do is to build capabilities in R&D. And in your introduction, you framed it very well with respect to, there is things where there is development plus research and development across all families. As one thinks through the idea of building long-term capability in this space, where should we invest our efforts? In the first ring light that you showed at the beginning, or in the expanded light where we really expand the search, or really in the fields where there is potential for anything to arise, especially for us who live on the continent, there is a lot of potential for other things to arise that were not on the radar. So how do we build capabilities? What do we focus on? And I don't suppose that that's a question that would necessarily come from political leadership. I think it's a question that ought to be addressed from a science leadership perspective. Thanks.
Thank you. Anna Maria?
Yes, and thank you for this question. It's excellent. It's allowed me to explain a bit more the idea that it's just a proposition that we hope will work to make better together. So the main point is that this basic research, including the surveillance, learning how to monitor our pathogens that are circulating in our backyard, understanding how the characteristics, how they change with the basic research to learn how to choose antigens and develop medical countermeasures is critical. We agree with you, and we are proposing this is done for the entire family, not for one pathogen. But on the other hand, we understand that to get the funding to maintain the interest from the policymakers and the politicians, you also have to work on pathogens that are relevant in your locality. So what we are saying is you have to work on the pathogens that are causing trouble now, that are known, the first ones, the priority pathogens. But you should do so with that eyes open on basic research for the prototype pathogens and for the unknown pathogens that are in the dark areas. It sounds complex, but with the technological advances of today, this is possible, and it can happen. And I think really with all my heart that that's the best way to achieve equity, that we really build this research capacity so that scientists from all around the world stand in conversations as equals, as contributors of the knowledge, the data, and the potential. And that's the best way to achieve equity in the short term and the mid-term.
It's going to be an evolving process, I think, that we're going to have to look at. But having a framework is going to be very helpful. Richard?
Yeah, just to add something, I mean, I guess I would counter a little bit that that would all come from the research space, and coming back to thinking about surveillance and data and understanding. A lot of that does need the political will to fund healthcare systems and to progress to universal healthcare, because our surveillance starts in communities, it starts at the lowest level of the healthcare system. and generating better data to understand risks and to help us prioritize has to start with that fundamental building block. So I worry a little bit that we end up thinking that everything is going to come from research, and we know that unfortunately pots of research money globally are dwindling. So there's less money to do all the things we want to do, but we have to keep the political pressure on to fund healthcare systems, to fund the surveillance that stems from that, and we have to see that as a fundamental thing that requires pressure on governments to stick to promises in the Abuja Declaration and other things.
Yes, and I mean, the one thing I would add to that, and Anna Maria, perhaps this is for you as well, is that I was recently attending a congress at Oxford University of their pandemic institute, and it was the second conference on pandemics, and there was a session of all the donors, the panel discussion. But I do think there needs to be more coordination between donors. It's not clear, for example, taking just Richard's example, wanting to do a sero survey here of high-risk populations for Mpox, just to see what, if there's been significant exposure or really not. It's really not clear who you go to. So I've knocked on the doors of several big donors. I'm not mentioning anybody, and some people are saying, no, it's not it's quite right. It's not what we do. It's not what we want to do. Others, yes. But I think, more coordination between the donors themselves. So for people who want to then do a sero survey, want to explore new product development, that it's clearer where you go to. And there needs to be, and as much as we're going to have the core, I think that we need to have more donor liaison and collaboration. So that becomes an easier one-stop shop for people who do have ideas. I don't know if anyone wants to comment on that. Anna Maria?
I totally agree with you, Helen. And because of that, there are two steps that we are taking with our colleagues. And I think the challenge, again, is the small groups and the small clubs for all of us. But there is GLOPIDAR, the Global Research Collaboration of different donors. They are going to have a meeting in December. It will be hosted by WHO. And one of the topics that is going to be coming out is how donors can consider supporting around the viral families approach. We hope that we will invite the core leaders to that meeting to present the concepts and etc. And the second is in collaboration with CEPI and other donors. There will be a small meeting of the bigger league donors, where we also are going to discuss the viral families and how they perceive the possibilities of collaboration, etc. But this works if we have this research and development roadmaps with collective identification of research priorities. This doesn't work if we all try to knock on the doors of every donor, but we already have a roadmap that is accepted, that research A, B, and D are the top priorities, are in the critical pathway for that viral family, then it will be better. And who will decide which ones are the priority research? The scientists deliberating at the center of the corps as a network, not us, the scientists. You will have talked about this, Helen, and I'm personally very committed to this idea of a decentralized scientific debate, because that is where science makes progress and discoveries are made. Thank you.
Thanks. So as we wind up, Ondari, do you want to have some last reflections on what you've heard in the discussion here?
I think my parting words will be, for us to be able to be ready for the next pandemic, we must, one, realize that no one cannot be able to do it alone. When you go back to 2020, or in January, it was found in one country, all of a sudden it was the entire globe. And also we are facing a continental health security issue right now, or a public health emergency with Mpox. And one of the discussions that is going on right now is about leadership, who should be able to be able to do that. And then the other one is about funding and financing that. At the end of the day, donors will have vested interest, but we're coming back to what Richard said. If indeed we took time even to the Abuja Declaration or the Algiers Declaration that was more into research, and we're able to generate the data, then we can be able to say, at least from what we do have or what is available, this is where we should be able to pivot towards as priorities. But ultimately realizing the entire value chain of vaccine manufacturing is an ecosystem that has to be approached holistically, and we must be willing, more so from external players, to leverage on already existing synergies and beyond solving the problem, to be able to look at sustainability. Thank you.
Thank you. Richard, last thoughts from you?
I mean, really just wrapping up what we've said. I mean, I think coming back to your point about coordination of funding and research funders and things. I mean, the other aspect there is the avoiding the short-termism and really looking at the long-term approach. And that's why the WHO approach and that framework is, I think, so helpful, because that's clearly a long-term approach to thinking about this problem. We all know that many research funders move between priorities. Even within a disease field, they'll move between prioritizing treatment research, prevention research, and that can't work. So we have to have the coordination, and as always in these discussions, the same key things come up: coordination, leadership, long-term commitment, and these are the things that we must challenge ourselves and challenge everyone to believe in and to deliver on.
Thank you. Anna Maria, any last thoughts from you as we close?
I think I am convinced, as the previous speakers, that we can solve these problems if we work together. There have been 3 things that have been identified. What is the strategic direction that we are proposing through the CORCs and the R&D roadmaps and the collaborative approach? The second is the political and the funding leadership. And we mentioned some mechanisms, but we should try to keep working together and believing in multilateralism. I know there is a temptation of me first, and that's a terrible sin. We should trust each other. And as my boss says, we are competitors. We have to admit that, but we also share a common goal. So we should go towards that common goal. And the third point, I think, is that it's a road that we have to walk together. We have a proposal for a strategy. We will have to work together to make it better every day, to refine it, to ensure it addresses the problems and produces the solutions we were hoping for. So I think, at least from the WHO perspective, we are open to walk this walk and to work with all these scientists and researchers and developers to make it happen, because it's very important for us.
Thank you very much. So unfortunately, Nikes Ndembi from Africa CDC wasn't able to join us, and Abdurrahmani Marufi also was pulled away. I think that people in this field are being pulled away because there's so much active dialogue going on. So I'm just left with one thought. I think this has been hugely helpful to hear what the initiatives are and to be given these different frameworks and initiatives. And that's that saying, you know, if you want to go fast, go alone. And if you want to go far, go together. So with that, thank you all very much. Thanks for your attention, and thanks very much to the excellent speakers. Thank you.
Goodbye.
Thank you so much, Prof. Rees, for chairing what really was an excellent session. What we had promised you in the session was that you will come out understanding what the global priority is around preparing for the next pandemic, since there's just so many viruses to think about. And I think that was certainly achieved. There's clarity around the priority families, priority pathogens, prototype pathogens, as well as prototype pathogen X. And so with that said, I'd like to thank our panel for sharing these very helpful insights. So we will now break for lunch. Lunch will be for about an hour. It will be at the room where we had the mayoral reception last night. When we come back, we will have 2 breakaway sessions in the rooms just outside of here. There will be one on regional regulatory mechanisms, chaired by Ms. Posha Nkambule from SAPRA. And then we will have a session on local manufacturing, chaired by Dr. Betty Dungu. So please, colleagues, enjoy lunch, and let's see each other at 1:30 in the breakaway rooms.